So welcome to the annual Bay Area Symposium on Viruses. So my name is Laurent Coscoy. I'm an associate professor in the molecular and cell biology department here at UC Berkeley. I'm also part of the chairing committee of the symposium. And I'm also the associate director of the Center of Emerging and Neglected Diseases, who's hosting the symposium today. And so it's a pleasure to have you all here today. This is the sixth year that we are running this symposium. And we are all here today to hear about exciting research and new development of virology. So we are lucky to have an excellent set of academic and industry speakers, as well as a group of competitively selected graduate students and postdocs that will be presenting as well. We have an exciting panel on Zika virus. And none of the least, we have also some exciting posters and receptions following the symposium. So we are, all interested in, understanding, preventing, controlling human, animal, plant viruses. Not that it's needed, but, it's an exciting time to do virology, and the news reminding us of that. Every year, and in particular this year, we all heard about the widespread of Zika, the world spread of Zika now, and the health concerns associated with Zika. We have all heard associated as well, for example, of this new oncolytic virus that has just been approved by the FDA to treat melanoma, which is an HIV-1 based virus. And so altogether, we share an interest in virology. And let me see. We share an interest in virology, but we also altogether live in an exciting place that is the Bay Area, where we do research and we live every day. Together, we are a community of shared passion, shared locations, and the purpose of today's conference is to bring together and further build this community. So make sure to take advantage of that. And, for example, during the lunch or the receptions, talk to people, and why not start a new collaborations? Obviously, as a community, we need to do more than meet only once a year for a symposium. So as you might know, we have set up a few years ago a platform that we have named BayViro, which is indicated here, that aims to further support us as a community. We want BayViro to feature all the Bay Area laboratories and companies that have an interest, that have an interest in virology. We want BayViro to highlight the funding opportunities that are of interest to us. We want BayViro to tell us if there is a new job opening in virology that might be important for postdocs or graduate students coming up. We also would like BayViro to be more than that, and allow us to exchange and take advantage of all our global expertise. For example, why not use a listserv associated with BayViro to ask questions such as, I'm interested in growing adenovirus. My lab doesn't have any expertise on it. Could you tell me which cell type I use? Can you give me a protocols? And we hope that people might jump in and exchange freely. Obviously, to further build this community, we need your support. And we need your help. And in particular, we need you to contribute. So, for example, if you hear novel funding opportunities, let us know, and we will post it on the website. If your lab is looking for a new technician, same thing, let us know. Or if you know a new exciting development that is happening at your university, tell us about it. Write a small article about it, and we will post it on the website. Share it with everyone. So keep the community going by continuing to be part of it, not only today. So we also have circulated an email very recently where we want your input about what you wish BayViro to feature and to have. So please look at this survey, fill out the survey, and you will have a chance to win a small gadget, okay? So I never heard about it before, So, but I'm sure my students will all contribute very, significantly to this, survey. So I don't want to take more of your times, but I want the symposium to progress. But I would like to thank several people. And not the least, Michael Eichberg, which is the executive SEN director, and Xyloses Lyons, which is on the back, which is running all the table there, which is a program manager of the Center of Emerging Neglected Diseases. And they work very hard to have the symposium work very smoothly. They've been contacting everybody. They're setting up Bevaro. They're taking care of the listserv. So they really deserve a big thanks from us. I also would like to thank all my colleagues from the committee, the chairing committee of the symposium, which is like Paul Lucy, Melanie Ott, Peter Sarno, and Phil Norris, who's here. So a big thanks for them. They've been really helpful in selecting all the talks, having the work that makes the symposium the success it is now. We also obviously want to thank the sponsors from which the symposium would not be possible and would not be running. In particular, Gilead, which is our platinum sponsor, has really stepped to the plate and really helped us a big deal. The Blood System Research Institute, BSRI, CFAR, and SAND have also significantly contributed to the symposium. So I would like to thank them all. And obviously, the one who's helping a lot as well are all our volunteers. All the students are sitting on the first row, that are sitting on the back, that will handling the microphones, and those deserve a big thank as well. So thank you all. Thank the community for coming out today and joining us. And we'll be starting the symposium now. And the first speaker will be Jim Hurley, which is a colleague of mine. He's doing excellent work on HIV, biogenesis, and how HIV manipulates the host cells. So the floor is yours.
All right, thanks. Good morning. I'd like to thank the organizers for giving me the chance to talk about our in vitro take on retrovirology that's perhaps going to be a bit different than the rest of the symposium. So the biogenesis of HIV particles is an essential final step in the HIV replication cycle. With no particle biogenesis, there are no progeny virions, there's no infection, and we think of this as an underexploited target for therapeutics. And I'm going to tell you about two aspects of this. One aspect is the release of the particle. HIV particles cannot release themselves. They have to co-opt the host escort machinery. In order to do this, the escorts act at the neck of the nascent virus. And so the first half of my talk will be about that. The second half will be about the question of how the HIV particle knows to package one copy of a dimeric unspliced RNA genome in the presence of a vast excess of cellular RNA. And that's work I've never presented before and which is unpublished. So why do this in the first place? Richard Feynman said, "What I cannot create, I do not understand." And I think that's reason enough to try to reconstitute these processes, but my corollaries of this are, if you don't have the structure, you can't understand the function, and what you cannot create, you cannot determine the structure of. So the ultimate goal of the reconstitution is not just to understand the workings of the machine, but to recreate it such that you can get structural information. So to go beyond determining structures of, say, static states of viruses and proteins, but to actually determine structures of snapshots of the process. So all of this starts with the HIV GAG protein, the structural component of the GAG of the HIV virion. GAG has 4 domains that are connected by flexible linkers. The MA matrix region is responsible for attachment to the plasma membrane through its binding to the lipid PIP2. The CA, the capsid domain, is responsible for the structure of the lattice. I'll be talking a lot about this. It's responsible for the assembly of hexamers and then the assembly of those hexamers in turn into a larger shell. The nucleocapsid domain is a zinc-containing domain that's responsible for selectively binding to and packaging genomic RNA. That will be the focus of the second half. And then P6 is responsible for the recruitment of the escort proteins, the focus of the first half of the talk. So PIP2, assembly, RNA, escorts. So to make any headway in this, we had to be able to purify GAG in biochemical amounts. This actually had not been done, and Lars Anders Carlson, who's in the lab, sitting right there with his HIV t-shirt. And by the way, Lars does own more than one shirt, but he's wearing this for the occasion. This shows the purification of a monodisperse peak of GAG. This is myristylated. So this is really the basis for everything else I'm going to show you, the access to this reagent. So the next step in this process is to come up with a lipid platform on which to assemble the GAG. And the platform of choice are giant unilamellar vesicles. These are vesicles that are as big as cells. They can be up to 100 microns across. And what we do is inject the proteins on the outside. So this is an inside-out budding process. The GAG protein forms shells that protrude inward, and this is mimicking the outward budding process from a human cell. And so this is an example of how this works. This GUV contains 5% PIP2, which attracts the MA domain. This one contains an equal amount of charge in the form of PS, and it doesn't attract. So the 5 versus 20% is because there is a -4 charge on PIP2, a -1 on PS. So these buds protrude inward, and this mimics a situation that you can actually see biologically in HIV virions that are defective in the PTAP sequence in the late domain. These virions get stuck on the cell surface. So you see a nascent viral particle connected by a narrow membrane neck to the rest of the cell, which cannot be released because the escort proteins are not engaged and the severing does not occur. So what are the escorts? They were originally discovered in endosomal sorting. That's where they get the name. E is for endosome. Escort stands for endosomal sorting complex required for transport. But they're required for much more than transport. They're required among certain archaebacteria in ancient evolution for cell division. And what the escorts do is they go to regions of negative membrane curvature and they sever membrane necks, and they can do this at the cytokinetic neck. They can do this in internal budding into endosomes. They can shed exosomes this way, microvesicles, and they can shed many viruses from the plasma membrane, and even in other cases, they can create viral replication compartments in the ER, and they can drive budding of viruses out of the nucleus. But we are going to focus on this plasma membrane event that's important for HIV. So during the 2000s, my labs and others really focused on understanding the structure and function of individual escort complexes. And by 2010 or so, we'd really worked through the whole list and turned our attention to integrating the structures rather than doing structures of individual complexes. So the idea is, can we assemble GAG, and then can we assemble escorts on top of that GAG platform? So we knew a lot from genetics about the escort cascade and the ordered recruitment. So there are a lot of these factors. Each one of these is a large multi-protein complex they need to assemble in order, and then the series of other individual proteins assembles on top of that, and all this is needed for the system to work. So this is in yeast, and we were able to mimic this through in vitro reconstitution. The in vitro side and the yeast genetics matched perfectly. So the situation was not so clear with the human proteins in HIV when we got into it. Wes Sundquist in particular had systematically knocked down human escorts and looked at effects on HIV bud release. And if you look at these highlighted components, these are ones that Wes was able to knock down. In some cases, other labs have looked at these also and found that that led to a block in HIV release. Now, if you look at the cognate proteins in yeast, some of the key elements in this chain of connectivity actually had little or no phenotype when they were knocked down in cells. And yet, in the biochemistry, these guys were found to interact one to another in the same chain of events. So there was a discrepancy between the in vitro binary binding studies and the knockdown phenotype. So what we decided to do is look at the in vitro system in a more sophisticated way and see perhaps in the setting of the membrane and the GAG, some of these things could be bypassed. So perhaps you don't really need all of these guys if you set the system up in a more sophisticated way. And what Lars did then was to purify all of the human proteins in this entire cascade. And many of these, again, are multi-protein complexes. They're all full-length, intact complexes. And then he would fluorescently label them one at a time and look at different combinations and look at what could be recruited. So here in this system, you are looking at GAG puncta, you are looking at labeled escort-1 complex, and you see nice colocalization. That's what's shown here. And if you mutate the PTAP on GAG, you reduce the colocalization. There is a second branch to this pathway. You can recruit the escorts to HIV buds independent of escort-1 through another protein called ALEX. And we can recapitulate that as well, that PRD is an autoinhibitory element, and consistent with the biology, we saw the recruitment go up when we deleted the PRD. If we mutate the ALEX binding site, again, we completely lose the colocalization. So now we can start to work our way downstream. CHIMP4B is an individual protein that is the major scission engine in this reaction. So it's the thing that forms a large assembly in the membrane neck and is probably the single most important component in driving the constriction and breakage of the membrane neck. And that can be recruited downstream. In this experiment, the escorts upstream, the escort-1 and the ALEX are not labeled. And you can look at CHIMP4B. So here this shows that the activated ALEX mutant, delta PRD, recruits labeled CHIMP4. Here what we can see is if we add escort-1 and then we add everything in the chain of connectivity between escort-1 and CHIMP4, we get recruitment. But now what we've done is left out escort-2. Escort-2 is one of the ones that had no phenotype in the knockdown studies. And when we wanted to know, could you bypass that in this more sophisticated system? And the answer is no. You can't bypass it. If you leave one component out, you get no recruitment of the CHIMP4. So this highlights this very systematic and painstaking study that Lars did, where he would either include all the components or leave out one at a time, and when he leaves out one, he gets no recruitment of the CHIMP4. And so this pathway goes further. CHIMP4 is not the end. We need these other things, CHIMP2 and 3 and 1, to recruit the VPS4 enzyme that takes everything apart at the end. And what we can see here is that to get CHIMP1, you need everything. You leave out just these couple of last components and you don't get that. So the conclusion of this set of studies is that you need the entire connectivity of the pathway. So everything that we learned from yeast genetics and from in vitro studies of the yeast escorts do appear to apply in this setting. And subsequent to this, another study has come out that does implicate escort-2 in the HIV budding. And a very elegant study came out from Harold Stenmark that looked not at HIV budding, but at cytokinesis. And many of the same conclusions had been stated in the literature for cytokinesis in human cells and for HIV release, that namely that escort-2 was not required, CHIMP6 was not required. This revisitation of this by Stenmark showed that if you do very careful and complete multiple knockdowns, you knock down both branches of the pathway. In fact, you do need every component. So I think now finally, after probably 8 years of discussion and debate in the field over this, I think we have now resolved that there is a unity to this pathway from yeast to humans. You need every one of these components to work together. Now, so let me say something about timing of this pathway. I've just told you about order and taken a more static view of this. What if you look at the kinetics in live cell imaging done by Sandy Simon, published a couple of years ago? You can see that over the course of this case, you can see about 7 minutes HIV is assembling in puncta on the surface of the cell. What happens if you monitor the recruitment of CHIMP4 is that it comes in a burst right at the very end. It's not being recruited gradually with the gag, but something happens at a certain point in this process. It's recruited in a burst, and then you get release. And we wondered if we could explain this in an in vitro setting. Our hypothesis was that as the gag builds up, at a certain point, enough builds up to create membrane curvature, and that is part of what triggers the recruitment. So Il-Hyung Lee in my group, in a collaboration with Jay Groves' lab, was able to nanofabricate pits in glass and assemble lipid membranes to mimic the shape of the HIV bud. So this is an AFM image of one of these nanofabricated pits, and Lee was able to show that continuous membranes could be formed that would cover these. And then we did kinetics, where we would add CHIMP4 in various combinations of upstream proteins. And if we add the appropriate set of upstream proteins to recruit the CHIMP4, we find that it is recruited within about 1 minute very selectively to these pits. So we think it is plausible that the rapid timing of this burst phase is in fact driven by the onset of membrane curvature. So we worked with Janet Iwasa at Utah, who's a professional biochemical artist, to come up with an animation of our current model of how the escorts do this scission. This is incorporating work from a number of labs in addition to ours that I didn't have time to go into. But here you see the gag assembling upstream escorts. This is CHIMP4, and we think CHIMP4, our current view, is that it makes a funnel that points outward and acts as a spiral spring that snaps the connection loose. So this is a hypothesis at this point. We're very involved in testing, but a neat idea. So now let me get to this unpublished story. I've never presented it before, so excited to get to show it and get your feedback. So a key aspect of the particle biogenesis is the packaging of 2 copies of the unspliced genome. So we set ourselves a challenge of trying to replicate this in vitro. So we're asking the question, in a purified system that's nothing but lipids, HIV gag, and RNA, can we replicate this? So this is a risky project, because if there are any other cellular factors that are needed for this, it can't possibly work. So Lars took a jump off the deep end to see if this would work. And what you're looking at here are several different RNA molecules. This is the 5' UTR, which is the 5' 365 or so nucleotides of the HIV genome that are known to be the major packaging signal. Here's RE, which is another HIV genome component that's involved in nuclear export, but not known to be especially important in packaging. And here, the RNA 378 is a random, it's an mRNA, I think, from one of the escorts, so it's just a random unstructured mRNA of the same size as a control. And here are myristylated gag puncta, and here are puncta of the fluorescently labeled RNA. And you can see the gag packages all of these RNAs with equal efficiency. It appears not to care. So this is in a system with half nanomolar of these RNAs and nothing else. There's some magnesium, there's physiological buffers, but there's no competitor RNA. In the cell, there's obviously a vast excess of cellular RNA. So what happens if you throw an unlabeled a random RNA, so mRNA? You get an equal amount of packaging of the 5' UTR. You completely blow off the labeled mRNA, and you sharply reduce the RRE. So this is what it looks like. The unstructured RNA is completely blown off by the competitor, while the 5' UTR is unaffected, and the RRE is somewhere in between. So the HIV gag has a structure on the membrane. It forms what is the immature lattice, and we know a lot about this structure from cryo-EM studies. So this is cellular tomography of the lattice. It looks like a spherical lattice with a bunch of holes in it. And we now, from work by John Briggs, was published late last year, we have an idea at up to 8 angstroms or so of what the capsid part of this lattice is, and that let us design mutants based on the structure that block the formation of each of the major symmetry elements, the 2-fold, the 3-fold, and the 6-fold symmetry axis. So here's what happens if you start to blow up the symmetry elements. You no longer get these sharp puncta. What you get instead is that the GAG diffusely coats the entire surface. So it's attracted to the surface by the PIP2 lipid in the GOV, but it is not forming puncta because it can't form the appropriate structure. And so this shows the reduction in binding if you compare wild type and you compare the mutants. So wild type is only in puncta, no diffuse. The 2-fold mutant is primarily diffuse, for example. None of these by themselves entirely block the puncta formation, but we can tell from this shift to the diffuse binding that the formation of the lattice is being screwed up. And so now the question is, if we add back RNA, can we rescue that? The RNA will grab the nucleocapsid domain and bring this back together. So can the RNA function as a glue for the GAG to restore the defect in the CA lattice? And the answer is largely yes. We get back this assembly. And so this is, again, with a half-molar 5' UTR. But this only works in the absence of the competitor. So now we've thrown in the competitor, and once again, it starts to become diffuse. So all of these structural mutants can be rescued by the specific RNA in the absence of the competitor, but only the wild type is robust to this, to the packaging and assembly in the presence of the competitor. So what this is all pointing to is that there's an intimate interplay between the ability to do the packaging right and the ability to do the assembly. If you screw up the assembly, you screw up the selectivity. You can rescue it, but you can't rescue it very well in a realistic setting where you have competing non-specific RNA. So I had mentioned that a key issue of this is that HIV can count. There's a dimeric genome that's packaged. So a dimerization is known to occur through a motif called the DIS, and Summers' lab has studied this in depth. They've shown that you can block dimerization by mutating this signal. So Lars constructed this mutant to block dimerization. We also know that flipped out G bases are important for the NC recognition. So the elimination of these 7 Gs reduces binding to NC, so we've eliminated those. And then finally, we know that splicing eliminates packaging. So we've started looking at some of these. These are ongoing experiments. And what we've found is that the monomerized sequence or this flipped out G reduced sequence reduce the selective packaging. And now we are starting to hit the limits of the dynamic range of this assay, but when we resensitize this by combining the 2 mutants, we can completely abolish the binding. So that's consistent with the idea that the simple in vitro system includes most, if not all, of the determinants for this packaging of the dimeric genome in this state. And then I just want to address one last point in my remaining few minutes, and that is, what is the real nature of the competing RNAs in the cell? So Paul Benesch published a paper in Cell, I think 2 years ago now, maybe a year and a half ago, where they did PAR-CLIP and they inventoried all of the cellular RNAs that interact with GAG at various stages in the life cycle. And the dominant RNA interacting with GAG through most of the path through until it's assembled and begins to be released are tRNAs. So those are the most abundant RNAs. So we thought that was really the most relevant thing to look at rather than looking at competition with mRNA. tRNA is also known to associate strongly not just with the NC domain, but it also interacts with the MA domain. So that same basic patch that binds to the acidic lipid can also bind to the acidic tRNA, and that tRNA binding can inhibit the interaction with the membrane. So the tRNA can compete two different ways with the proper function. So you might think that this makes life really difficult for GAG. So here's what happens if you throw in 1 micromolar tRNA. You can knock all of the GAG off lipids. And so just to cut the story short, if in the presence of no tRNA, if we look now in a bulk assay, we've switched to a different lipid system. I can go into it in questions if you want. We can come up with a system where there's really no specificity in binding. And then what we find in the presence of now a massive excess of tRNA, 20 micromolar, so perhaps a realistic model of the massive excess of this in a cell, we can get a large enhancement in the binding of the 5' UTR, but now we've gone to a new construct. We've gone to what we call the HIV-1947, which is the first 1947 nucleotides of the HIV genome, so purified in biochemical quantities, fluorescently labeled. That has a massive increase in specificity in the presence of the tRNA compared even to the 5' UTR. So here's the model for what's going on. We think there is a subset of the GAG hexamers, so perhaps some number, 6, the ballpark of 6 hexamers out of the much larger number in this lattice that are forming a structurally specific, a stereospecific binding site for this 5' UTR, but that in addition, there are bulk effects, so a less specific binding to the NC through tail regions that don't individually contain a packaging signal, yet through their bulk and charge also contribute to binding. So what's going on with the tRNAs? The tRNAs are buffering all of this. So here, those are these little V shapes here. And the idea is that these little V shapes can bind to both the membrane binding site and the RNA binding site on the GAG and then buffer them and prevent them from assembling with any RNA that is not good enough, that they are in a way helping to purify this and select for the correct genome. So in summary, what was done in the study is that selective RNA packaging was reconstituted from a completely purified system with nothing but lipids, pure proteins, and RNAs. There are no cell extracts or no unknowns in this system. We showed that the lattice is intimately interconnected to packaging specificity in work I didn't have time to show with Jennifer Doudna and Yun Bai. We showed that this is mediated without gross changes in the structure of the RNA, so that this is due to a recognition of structural motifs that are already understood, and that the tRNA in the cell strongly inhibits genomeless packaging and helps create the specificity. So that the tRNA, paradoxically, the competition is actually essential to drive the correct process, although it might at first seem like something that would inhibit it. So overwhelmingly, the majority of this work was done by Lars. So here he is pictured in this shirt. That's why I had to tell you he does own other shirts. And then in addition, most of these people I mentioned as we went. So I will stop at this point and take questions. Thank you.
I'm curious about whether or not, I was trying to figure out the order of things in my head. So from the first part of your talk, I gather that GAG can assemble at these membranes in the absence of RNA. Is it not forming lattices at that point? Does RNA interaction help with the actual lattice formation? And does what you've shown in the second part shed any light into how the number of RNA molecules are selected for packaging?
Yeah, so I presented the two halves of the talk in the order in which we did them, which is the opposite order, of course, as to what actually happens in the virion biogenesis. And just for full disclosure, I will tell you the real reason we did all this. So my real passion is understanding the escorts and the membrane severing. I'm not an RNA guy. We got into the RNA because I showed you we very successfully reconstituted this whole cascade. But at the end, we did not see these buds being released into the interior. So we thought, what is missing? And the most obvious missing thing is the RNA. So we spent a lot of effort now in getting that right. Now that's done, and we're going to go back and see, will that improve the lattice formation enough to drive the correct budding into the interior? So the short answer to your question is, I think in the first half of the talk, we think some of the lattice is being assembled right, but it's not correct enough to be competent to bud. So partly right, partly not. Yeah.
Great talk. So why do you think that it's just 2 genomes that are packaged as opposed to
So
4?
what we know is that this dimerization signal is important for binding. Now that doesn't tell us that in vitro we're not getting 4, 6, or 8. So that's a really interesting question. There is room sterically for more copies than are actually packaged. So this question of how HIV counts is, say, unresolved, a fascinating question. High up on our radar, but we don't know. Someone in the back who seems eager to. Do you have a question back there?
Yeah. So great talk. So I know that some of the frequently packaged RNAs within the particle are 7SL and ALU elements. Have you sort of used those as markers too to see if they're tapping into the same things that your HIV constructs are, versus are they sort of being packaged in the same way? Do they compete differently than?
I actually didn't get the specific name of the RNAs.
It's like 7SL, for instance.
No, we haven't looked at that. I'm interested in that one for other reasons, but it's a good suggestion.
Are you packaging icosahedra or flasks? Do you get the characteristic shape of the HIV particle?
So this shape is an imperfect sphere. It's not an icosahedron. It's much bigger. There are probably something like 2,000 copies of GAG, and because these are hexagons, this can't close into a sphere. So it's a defective sphere, I think, is how I would characterize it. It's not an icosahedron. So we don't think it's flat. In some of these images, we can see things that seem to protrude into the lumen, but these GOVs are really intractable for electron microscopy.
Okay, but for HIV, the actual particle is flask-shaped. You don't get that.
Oh, is flask-shaped? I thought you said flat. That's the mature particle. So after the proteolytic processing of GAG, we are looking at the immature particle. There is no HIV protease in our system, so it cannot mature. We're interested in looking at a maturable system in the future, but we haven't done that yet.
Okay. Thank you.